Hello everyone, how is your day going? Welcome again to Ferdinand tutorials. Today we want to answer about 101 past question 2019-2020 session. The other time we did 2020/2021, so we're doing this today. Now they gave us 6 questions here and they said that we should choose any 4, but I'm not going to be answering any 4. I'm going to be choosing the 6 of them, so that in case you see any in exam, you can be able to answer it speedily and without any problem. So let's go. Now the first question, it says, in not more than 2 sentences, define the following: genotype, phenotype, homozygous, heterozygous, double recessive, and alleles. Now we say that genotype, this is the genetic constitution of an organism. Now I gave an example, R capital R. And phenotype, we say that this is the outward expression of an organism. That is what you can see. Example is the long stem, you understand, and the genotype is capital T, capital T. Now let's go to homozygous. Now this is when an organism contains identical genes. Example is the capital T, capital T. Meanwhile, heterozygous, this is when an organism contains 2 dissimilar genes. Example, capital T, small t. Are you there? Now, double recessive, this is when an organism is homozygous for a recessive gene. Example, small t, or small a. Number VI, we say alleles. These are alternative forms of a gene that arise by mutation and are found at the same place on the what chromosome. That is that for number 1A. Now, let's answer number 1B. Number 1B, it says, what are the genotypes of the following? We say Klinefelter syndromes, Turner syndromes, blood group B, blood group AB. Now, first off, I say Klinefelter syndromes. Now, when you hear about Klinefelter syndrome, it is a genetic condition in which a male is born with an extra copy of the X chromosome. Now, remember that every male has an XY chromosome. That's what makes it a man. You understand? So, but when we have an additional X chromosome, now the new genotype in Klinefelter syndrome is now XXY, because we already have an XY in the male chromosome plus the addition of the extra X, which is what? XXY. That's for the Klinefelter syndromes. Now let's go to the Turner syndrome. Now Turner syndrome is a chromosomal disorder in which a female is born with only one X chromosome. Someone will ask you, what's the chromosome or what's the genotype of every female? You begin to see that it is what? XX. Now here, it is a chromosomal what? Disorder in which a female is born with only what? One X chromosome. Thus, the genotype in the Turner syndrome is the what? X chromosome, or the X genotype. Now, number III, the genotype for blood group B is always B. You understand? Here we say IB in homozygous condition, and what? IB small I in heterozygous condition. Meanwhile, genotype AB is always what? AB. Blood group AB, the genotype is what? AB. Now, for question 2A, let's get the past question. It says, protozoa have, now look at the option, short leg generation in time, short or long generation time, high or low reproductive rate, large and small surface area to volume ratio, small or large size. Say choose one underlined option. Now here I chose that protozoa have a short generation time, protozoa have a high reproduction rate, protozoa have large surface area to volume ratio, and protozoa are small in size. Now number 2B, let's look at the question in number 2B. It says, why are protozoa considered more advanced than cells of multicellular organisms? Now I answer that this is because none of the cells of the multicellular organisms have adopted multicellular somatic organization characteristics of metazoan cells or metazoan organisms. Now instead, protozoa have developed relatively complex subcellular features. We're looking at the membranes and organelles here, which enable them to survive the rigors of their environment, the challenges of their what? Of their environment. Now question 2C, it says, what are the 2 forms of asexual reproduction in Porifera? Now here I say that we have fragmentation and what? And budding. That's the 2 forms of asexual reproduction in Porifera. Now number 2D says, list 4 advances of Platyhelminthes over colentrates. 4 advances. I started with Platyhelminths are tripoblastic animals. When you hear about triploblastic, that means they have the 3 germ layers. We have the endoderm, we have the ectoderm, and we have the what? Mesoderm. That's platyhelminthes. While coelentrates are diploblastic, that's the endoderm and the what? Ectoderm alone. Now reproduction is majorly sexual in what? Platyhelminthes, but asexual in what? Coelentrates. Now, nervous and circulatory system is present in platyhelminthes, but absent in coelentrates. Platyhelminthes are bilaterally symmetrical, while coelentrates are radially what? Symmetrical. Now, number 2E, question 2E, it says, in 2 sentences, describe the morphology of mollusks. In 2 sentences, describe the morphology of mollusks. We are talking about morphology, we are looking at the form. Are you getting it? Form of what? Of an organism. Now, mollusks have soft bodies consisting typically of an anterior head, a ventral foot, and a dorsal visceral mass. The body is more or less surrounded by the fleshy mantle, which is an outgrowth of the body wall and is commonly sheltered in an external limy shell. I know you love the video you're watching, so please just click on the subscribe button. That's a way to support what we're doing here. Like this video as you're enjoying it right now. Like it, comment your questions, some of the things you've watched so far. I know you're not done yet, but some of the things you've watched so far, comment your questions and share this video to your friends. Please, this is a way to support us. So let's go back to the video. Number 3A, the question says, why in one sentence, why is mitosis regarded as equational division and meiosis as reductional division? Now we say that mitosis is regarded as equational division because the number of chromosomes in each daughter cells are equal to that of the parent cell after replication, right? We answered this question last time. While meiosis is a reductional division because the number of chromosomes in the daughter cell is reduced to half the number of the what? The parent cell. Now to the 3B question. Now to 3B, it says define hypotonic, isotonic, and hypertonic solution. Now let's go. We said that hypotonic solution, I gave 2 definitions, but I'll be going with the second one. I said this is when a solution in the surrounding has a lower solute concentration as compared to the solute concentration inside the cell. That is for hypotonic. Now for hypertonic, I said that this is when the surrounding solution has a higher solute concentration compared to that inside the cell. Now, meanwhile, the isotonic, this is when the solution has the same concentration of solute in both the surrounding solution and the what? Solution inside the cell. That is for 3B. Now, for 3C, the question says, state the primary function of the following. We have the cell membrane, we have the nucleus, we have the vacuole, and we have the what? The lysosomes. Now, we'll continue. I will say that cell membrane. Now, this is a semi-permeable membrane that regulates the exchange of substance between the cell and its surroundings. Please understand this thing. It's very, very important. I hope you are getting. Understand them. Don't just read and cram. No. Understand them because it will be easier for you to write in the exams. That is it for the cell membrane. Now let's go to the nucleus. Now we say that nucleus, it contains the hereditary materials, that's DNA, which control all the activities of the what? Of the cell. Now the vacuole, it helps in the storage of various substances, including what? Waste product. Now it also helps in what? Osmoregulation. Meanwhile, IV, lysosomes, now they carry and distribute enzymes which are used for digestion of foreign bodies and worn out parts of the cell. Now in some cases, we say that lysosomes secrete what? Lysozyme. Now what does lysozyme do? Now lysozyme kills or destroy what? Senile cells. That is by the way. Now let's go to 3D. 3D question. Now 3D says, it says, name the 2 specialized cells involved in each of the following: translocation and transportation in plants. Now I want to say something. Both translocation and transportation, they are all that was transportation in what? Plant. So the 2 specialized cells there are what? Xylem and phloem. So when this kind of question comes in exam, under translocation, write it as xylem and phloem. Under transportation, also xylem and phloem. Meanwhile, we say that translocation is the movement of materials in plants from the leaves to the other parts of the plant. Thank you. Now let's go to the number 4A. Now 4A, what's the question? It says, give 5 comparisons between the alternation of generations in bryophytes and what? Pteridophytes. Now we said that bryophytes are non-vascular. Now remember, when we're talking about comparison, we are comparing. That means you are bringing the similarity and you are bringing the what? The differences. So we said that bryophytes are non-vascular while pteridophytes are vascular. Bryophyte has no vascular tissue, while pteridophyte possess vascular tissues. Bryophyte cells are haploid, while pteridophyte cells are diploid. Now I come with to the similarities. I say both plants consist of the heteromorphic alternation of generations. They are multicellular sporangia. The corticles is present in both plants. That is for number 4A. Now, meanwhile, for number 4B, we state 5 characteristics of fungi. State 5 characteristics of what? Of fungi. Now, remember that fungi are what? Are microorganisms. Now, we say that first one, fungi reproduce by means of spores. Fungi is the phenomenon of alternation of generations. Examples include, we have the mushrooms, the one you eat, we have the moths found in your bread, and we have the what? The yeast. Now, some fungi are parasitic and can infect the host. Fungi store their food in the form of what? Starch. Now, the mode of reproduction is sexual or asexual. That is it for that. Now, we move to the next question. That's number 5A. Now, number 5A says, explain the following terms. We have the complex or conjugate protein, glycosidic bond, and name 2 types of glycosidic bond, essential what? Amino acids. Now we'll go that conjugate protein are protein made up of a non-protein group, also called the prosthetic group, attached to the protein part. Right? Now, meanwhile, glycosidic bond is a type of covalent bond that joins a carbohydrate molecule to another, which may or may not be another what? Carbohydrate. You remember in the formation of starch, we say that we have different monosaccharide units combined together, you understand, to form that starch. Now, when we're talking about different monosaccharide units, we're looking at maybe glucose plus glucose plus glucose. So they don't just join together. They join by their what? Alpha what? Glycosidic bond or beta glycosidic bond. So we have the 2 types of glycosidic bond here. We have the alpha and the what? The beta glycosidic bond. Now we have the essential amino acids. Now this is an amino acid that cannot be synthesized as from the scratch by the organism fast enough to supply its demand and must therefore come from the diet. That is it. Now the 5B. The 5B question is, it asks, it says, explain the classification of biocatalysts based on the reaction they catalyze. I gave you a simple acronym, OTHLIL. O for oxidoreductases, T for transferases, right? H for hydrolysis, L for what? Lyases, I for isomerases, and L for what? Ligases. Now look at oxidoreductases. They catalyze the transfer of electron from compound to another. Transferases, they catalyze group transfer reaction. Hydrolysis, they catalyze hydrolysis reaction, right? Now lyases, they catalyze addition of group to double bonds or the reverse. Now isomerases, they catalyze transfer of groups within molecule to yield what? Isomers. Ligases, the enzyme catalyze bond formation. We have C to C, CO, CN, we have CS, by condensation reaction coupled to what? Hydrolysis of high energy compound. Now 5C, this was the same question that they asked us last year. They're repeating it again. They say briefly explain the designation P680 and what? P700. And I told you that photostem 2 is known as what? P680, while photostem 1 is known as what? P700. P680 because it absorbs light best at 680 nanometer, and P700 because it absorbs light best at what? Best at what? 700 nanometer. Now the 6B question, it says, they ask us is a diametrically differentiate between prokaryotic cell and eukaryotic cell. That one you can draw it on your own. Plant and animal cell. Plant cell as a prokaryotic and an animal cell as what? A eukaryotic. Now these 5 groups of microorganisms, right? These 5 groups of microorganisms, we have the bacteria, we have the viruses, we have the archaea, we have the fungi, we have the what? Protists. Number 6C, it says classify microorganisms based on I, oxygen requirements, temperature requirements, pH requirements, and then what? Nutritional requirements. Now let's go for the first one. Oxygen requirement, we have the obligate aerobes, microaerophiles, facultative anaerobes, obligate anaerobes, strict anaerobes. All right. Now temperature requirement, we have the psychophiles, that's cold loving microbes, mesophiles, moderate loving, thermophiles, heat loving. You understand? Now to the next one, which is the what? pH requirement. Now under the pH requirement, we have the acidophiles, they grow best at a pH less than 5. Neutrophiles, optimal pH, right? Alkalophiles, pH greater than 9. And meanwhile, nutritional requirements, we have the autotrophs and we have what? The heterotrophic what? Microbes, those that produce their food on their own and those that depend on other organisms to produce their own. So that is it. Thank you so much for watching that video. I hope you enjoyed it. I hope you loved it. So as I normally say, share this video. It's a way to tell others, it's a way to inform others of what we're doing here. That is if you like what you just watched. So share this video, click the subscribe button if you've not subscribed, like this video, and comment your questions, comment your suggestion. What other video do you want us to make for your consumption? This is the time to tell us. So tell us and we'll make the video. See you again when next we upload the video. Bye for now.